Hi everyone. In the previous chapter, we looked at the basic mechanisms of endocrine regulation and its partnership with the nervous system. As it will be repeatedly demonstrated, the nervous and endocrine systems often act together to regulate physiology. So endocrinology, though, is the study of hormones, the receptors, and the intracellular signaling pathways that they invoke. And so we have distinct endocrine organs that are scattered throughout our body. And these organs are largely, or at least, famously devoted to the secretion of hormones. So no introduction to endocrinology would be complete without looking at the endocrine glands themselves. And here's your map or study guide of learning objectives, basically learning the location and anatomical components of the pituitary gland. We're going to learn to list the hormones of the adenohypophysis and describe the general functions and identify the primary location of the target cells on which the hormones of the adenohypophysis act. We're going to identify the actions of the hormones that are stored and released by the neurohypophysis, and we will also discuss the structure, location, functions, and hormones of the pineal gland. The pituitary gland, or the hypophysis cerebri, is often called the master gland. It's a pea-sized gland located at the base of the skull in a bony pocket that's called the hypophyseal fossa, which is a bony depression in the sphenoid bone in an area called the sulcus turcica, or the Turkish saddle. You might remember that from last semester. It hangs from a stalk on the underside or ventral brain and is connected to the hypothalamus. Hormonal messages from the hypothalamus of the brain communicate with the anterior or front portion of the pituitary gland. The pituitary stalk, or infundibulum, has a unique arrangement of capillaries and veins known as a portal system, which allows the hypothalamic hormones to access the anterior pituitary without circulating through the body. There are two main components of the pituitary gland: the anterior pituitary, which is also called the adenohypophysis, and the posterior pituitary, or the back portion, which is known as the neurohypophysis. The posterior pituitary, or neurohypophysis, is in effect a projection of the hypothalamus. It does not produce its own hormones, but it stores hormones and releases the hormones that have been created by the hypothalamus. And the hormones travel down a nervous pathway to the neurohypophysis, whereas in the adenohypophysis, the anterior pituitary produces and secretes its own hormones, and hormones from the hypothalamus will travel through that portal blood system to the adenohypophysis, and we'll see how these are hormones that will kind of prompt the adenohypophysis to synthesize and secrete its own hormones. Here we see a diagram of the hypothalamus and its connection to the pituitary gland. The almond-sized hypothalamus is located below the thalamus and sits just above the brainstem. Have you ever noticed how we compare all the sizes and shapes of parts in our body to food? But I think that's actually more useful than when the book tells you, that a gland weighs 500 grams and is, 8 centimeters by 15 centimeters by whatever. You need to know that if you're a radiologist measuring things, but for most of our purposes, probably comparing to food is more useful. So anyway, all vertebrates have a hypothalamus, and its primary function is to maintain homeostasis, or the stability of the internal milieu or environment of the body. The hypothalamus links the nervous and endocrine systems by way of the pituitary gland. The hypothalamus will secrete, releasing and inhibiting hormones that will stimulate or inhibit productions of hormones in the anterior pituitary. And specialized neuron clusters, or neurosecretory cells in the hypothalamus, will produce hormones. Antidiuretic hormone, or vasopressin, and oxytocin are hormones that are made in the hypothalamus, and they will be transported to the neurohypophysis, where they will be stored for later release, and they're transported down a nervous tract. And one of the differences between the adenohypophysis and the neurohypophysis is the way the hormones make it from the hypothalamus into the pituitary gland. So in the neurohypophysis, those two hormones travel down the nervous system tracts. And in the adenohypophysis, the stimulating or releasing hormones will travel through that hypophyseal portal blood vessel. Now, the bulk of the adenohypophysis is known as the pars distalis, and this tissue is composed of winding cords and epithelial cells that are flanked by vascular sinusoids. And when we look at these in sections that are stained with our H&E or hematoxylin and eosin dyes, we see three different distinct cell types. We have acidophils, which have cytoplasm that stains red or orange, basophils that have cytoplasm that stains a bluish color, or the chromophobes, which have cytoplasm that barely stains at all or stains very poorly. The acidophils will contain cells that are the polypeptide hormones. These will include somatotropes, which produce growth hormone, somatotropin, lactotropes, which will produce prolactin. The basophilic staining cells will contain cells that have the glycoprotein hormones in them. And these are the thyrotropes that produce thyroid-stimulating hormone, the gonadotropes, which produce luteinizing hormone and follicle-stimulating hormone, and the corticotropes that will produce adrenocorticohormone, ACTH. It's the high carbohydrate content of the hormones within these cells that also stain bright purple on PAS stains. Here you can see a staining of a section of the adenohypophysis, or the anterior pituitary. There are 5 types of cells in the adenohypophysis: somatotrophs, prolactin cells, corticotropic cells, thyrotropic cells, and gonadotropic ones. Corticotrophs produce ACTH and the other hormones which are derivatives of pro-opiomelanocortin. Thyrotropic cells secrete thyroid-stimulating hormone, and gonadotrophs produce FSH, follicle-stimulating hormone, and LH, luteinizing hormone. Somatotrophs comprise almost 50% of the pituitary cells of the anterior pituitary. They are located predominantly in the lateral wings of the pituitary gland. And they, of course, produce growth hormone, also called somatotropin, or human growth hormone. Growth hormone is a protein hormone, about 190 amino acids, that is synthesized and secreted by the somatotropes, and it's a major participant in control of several complex physiological processes, including growth and metabolism. Growth hormones also have considerable interest as a drug used in both in humans and animals. And we'll talk a little bit how some people use it as their search for the fountain of youth. A critical concept in understanding growth hormone activity is that it has 2 distinct types of effects. First are its direct effects that are the result of growth hormone binding its own receptor on target cells, such as fat cells, adipocytes, for example, will have a growth hormone receptor. And growth hormone stimulates them to break down triglyceride, and it will suppress their ability to take up and accumulate circulating lipids. Indirect effects are mediated by insulin-like growth factor 1. This is a hormone that's secreted from the liver and is known as a somatomedin, a group of proteins that are produced by the liver when growth hormone acts on target tissue. A majority of the growth-promoting effects of growth hormone is actually due to IGF-1 acting on its target cells. So keep this in mind, and we can discuss the major roles of growth hormone and its little minion there, IGF-1, in physiology. So growth hormone promotes linear growth via the stimulation of protein synthesis and increasing the transport of amino acids through the cells. And most of this is thought to actually be an indirect effect through IGF-1. IGF-1 appears to stimulate the proliferation of chondrocytes or cartilage cells, resulting in bone growth. The growth hormone itself does not seem to have a direct effect on bone growth in stimulating differentiation of chondrocytes. And we'll talk a little bit about linear bone growth and when you have too much growth hormone resulting in gigantism as a disorder in children. The effects of growth hormone in a dysfunctional or disease state in adults is a little bit different, and that's known as acromegaly. We'll discuss that later. Other effects that growth hormones has through these somatomedins or IGF-1 are listed below. It'll stimulate cell division and cell proliferation. It can enhance T cell proliferation. It will increase the absorption of calcium ions from the intestines and will decrease loss of these from urinary excretion. Also, it will stimulate the hepatic glycogenolysis to increase the presence of glucose in the blood or increase glucose blood concentration. And you can always think of growth hormone in that sense as being a counter-regulatory hormone to the effects of insulin. Growth hormone will also increase the growth in soft and skeletal tissues and increase the uptake of non-esterified fatty acids by the muscles. Growth hormone has important effects on protein, lipid, and carbohydrate metabolism. In some cases, a direct effect of growth hormone has been clearly demonstrated, while in others, it operates through IGF-1. And in some cases, it appears that both these direct and indirect effects are at play. Regardless, in protein metabolism, growth hormone stimulates protein anabolism in many tissues. This effect reflects increased amino acid uptake, increased protein synthesis, and decreased oxidation of proteins. Concerning fat metabolism, growth hormone will enhance the utilization of fat by stimulating the breakdown of triglycerides, as we just discussed. And growth hormone is one of a battery of hormones that serves to maintain blood glucose within a normal range. Growth hormone is said to have an anti-insulin activity, or is called a counter-regulatory hormone, because it suppresses the abilities of insulin to stimulate the uptake of glucose in peripheral tissues and enhance glucose synthesis in the liver. Generally, the test questions that you're going to be concerned with is knowing that growth hormone is anabolic. It helps increase linear growth and muscular bulk, anabolic hormone. And also remember that it shifts all chemistry away from the breakdown of glucose and toward the breakdown of fats instead. And it does this in order to increase the blood glucose level, functioning as an insulin antagonist or a counter-regulatory hormone to insulin. Prolactin is a single-chain protein hormone that's closely related to growth hormone. It is secreted by lactotrophs in the anterior pituitary. The major target organ of prolactin is the mammary gland. It stimulates mammary gland development and milk production. And note that test questions will often try to trick you on this into choosing that prolactin causes the milk letdown reflex. And that is done by oxytocin. So don't be fooled by this. So prolactin develops the breast and helps with the development of the milk in it, but actually initiating the breast to push out the milk is the job of oxytocin. And keeping track of our neuroendocrine interactions, dopamine serves as the major prolactin inhibiting factor or brake on prolactin secretion. So dopamine is secreted into that portal blood by the hypothalamic neurons, and it will bind to receptors on the lactotrophs and inhibit the synthesis and secretion of prolactin. So agents and drugs that interfere with dopamine secretion or receptor binding will lead to the enhanced secretion of prolactin. And drugs that do this are a lot of your antipsychotic drugs. So you'll find sometimes people on antipsychotics will have larger development of their breasts and also produce milk. And yes, antipsychotics will cause male breast growth. And you can imagine this is the number one reason why men will fail to comply with their antipsychotic treatment. You might recall that tropic hormones are hormones that have other endocrine glands as their target. Most tropic hormones are produced and secreted by the anterior pituitary. These include thyroid stimulating hormone, adrenocorticotropic hormone, ACTH, luteinizing hormone, LH, and follicle stimulating hormone, FSH. The hypothalamus also secretes tropic hormones that target the anterior pituitary, and the thyroid gland in turn secretes thyroxine, which will target the hypothalamus, and therefore can also be considered a tropic hormone. So let's look at these four principal tropic hormones, beginning with thyroid stimulating hormone, TSH. It's also known as thyrotropin or thyrotropic hormone, but usually in common parlance, everybody refers to it as TSH. It is a pituitary hormone that stimulates the thyroid gland to produce its functional hormones, thyroxine, T4, and triiodothyronine, T3, which stimulate the metabolism of almost every tissue in the body. TSH is a glycoprotein hormone produced by the thyrotrope cells in the anterior pituitary gland, which regulates the endocrine function of the thyroid gland. The hypothalamus in the base of the brain produces thyrotropin-releasing hormone, TRH, which will stimulate the anterior pituitary gland to produce TSH. Later on, we will examine this whole TRH, TSH, T3, T4 negative feedback loop. Very important to learn. Adrenocorticotropic hormone, ACTH or corticotropin, stimulates the adrenal cortex. More specifically, it stimulates secretion of glucocorticoids such as cortisol and has little control over secretion of aldosterone, the other major steroid hormone in the adrenal cortex. ACTH is secreted from the anterior pituitary in response to corticotropin-releasing hormone secreted by the hypothalamus. It is secreted in response to many types of stress, which makes sense in view of the stress management function of glucocorticoids. As its name implies, follicle-stimulating hormone, FSH, stimulates the maturation of ovarian follicles in women. Administration of FSH to humans results in superovulation, or development of more than the usual number of mature follicles, and hence an increased number of mature gametes. FSH is also critical for sperm production. It supports the function of the Sertoli cells, which in turn support many aspects of sperm cell maturation. In both sexes, luteinizing hormone, or LH, stimulates secretion of sex steroids from the gonads. In the testes, LH binds to receptors on the Leydig cells, stimulating synthesis and secretion of testosterone. The theca cells in the ovary respond to LH stimulation by secretion of testosterone, which is converted into estrogen by the adjacent granulosa cells. In females, ovulation of mature follicles to the ovary is induced by a large burst of LH secretion known as the pre-ovulation LH surge, and we'll learn more about that in our next chapters on reproduction. LH and FSH are called gonadotropins because they stimulate the gonads, in males the testes and in females the ovaries. They're essential for reproduction, but not essential for life. The principal regulator of LH and FSH secretion is the hypothalamic hormone, gonadotropin-releasing hormone, GnRH. The physiologic effects of hormones depends largely on their concentration in the blood or extracellular fluid. And almost inevitably, you will find that when we have either too much or too little hormone, we're going to have disease resulting. Precise control of our circulating hormones is therefore vital. So let's look at how we achieve this using our thyroid system through our hypothalamus pituitary thyroid axis. Now let's first talk about the concentration of hormone as seen by the target cells in whatever the target organ is determined by three basic factors. The first being the rate of production. So we're going to need control somehow over the synthesis and secretion of hormones, and this is amongst the most highly regulated aspect of endocrine control. Such control is usually mediated by positive and negative feedback circuits, and as you know, negative feedback circuits are more common in the body. We're also going to control the rate of delivery of hormones, and this would be by like the rate of blood flow to a target organ or the group of target cells. High blood flow is obviously going to deliver more hormone than low blood flow. And there will also be another factor, which is the rate of degradation or elimination of these hormones. So hormones, like every biological molecule, is going to have a characteristic lifespan or rate of decay, and they will be metabolized and excreted from the body. So we can shut off a hormone that has a very short half-life. But if the hormone has a long half-life, its effective concentration is going to linger for a whole lot longer, even after synthesis and secretion will cease. So negative feedback is the way most of these systems operate. Negative feedback occurs when the output of a pathway inhibits the input to that pathway. And we classically liken this to the home heating system, which is apropos for thyroid hormones. The heating system in your home is a negative feedback circuit. When the furnace produces enough heat to reach the set point on the thermostat, then the thermostat triggers the shutoff of the furnace, or it's feeding back negatively on the source of heat. So when the temperature drops back below another set point and the negative feedback is gone, the furnace will come back on. So feedback loops are used extensively to regulate the secretion of hormones in the hypothalamic pituitary axis. And probably the key example of this negative feedback loop is seen in the control of thyroid hormone secretion. The thyroid hormones thyroxine and triiodothyronine, T4 and T3, are synthesized and secreted by the thyroid glands themselves, and they will then affect metabolism throughout the body. The basic mechanisms for control in this system are illustrated on the next slide. So let's look at this. Here we have neurons in the hypothalamus secreting thyroid releasing hormone, or TRH, which will stimulate cells in the anterior pituitary to secrete thyroid stimulating hormone, or TSH. TSH will then bind to receptors on the epithelial cells in the thyroid gland. This stimulates the synthesis and secretion of the thyroid hormones T3 and T4, which affect basically every cell in the body. When blood concentrations of the thyroid hormones increase back to that set point or threshold, then TRH secreting neurons in the hypothalamus are inhibited and they stop secreting TRH. This is an example of negative feedback. Inhibition of TRH secretion also leads to shutoff of TSH secretion, which in turn will lead to shutoff of thyroid hormone secretion. As thyroid hormone levels decay below that set point or threshold, then negative feedback is relieved and TRH will start again secreting, leading to further TSH secretion. You can also see that there are both long arm and short arm negative feedback back on both the hypothalamus and pituitary. The pituitary is not just inhibited through the hypothalamus, rather also the increase in the level of T4 and T3 will directly feedback as well on the pituitary itself and stop it from releasing thyroid stimulating hormone, which will further stimulate that thyroid. Okay? Pay good attention to trying to understand that negative feedback loop through the thyroid. It is a classic negative feedback loop, and it's probably the most tested. This is one of those things that you will get tested on frequently in your board exams, and you will also see it clinically. And, some of you might end up actually in your lifetime get affected by some dysfunction in this negative feedback system. And so this is a way that you will learn how to understand your thyroid hormone tests. What happens when your T3 and T4 are high? Would you expect your TSH to be suppressed, or would you expect it to be high? What happens if it's not like what you expect? So you can start thinking a little bit about what you would expect from your blood tests in yourself and in your patients. The key to understanding the endocrine relationships between the hypothalamus and the anterior pituitary, or adenohypophysis, is to appreciate as we've already seen, the anterior pituitary is under strict control by the hypothalamic hormones. And these hypothalamic hormones reach the anterior pituitary through the hypothalamo-hypophyseal portal system. Quite a mouthful. A branch of the hypophyseal artery will ramify into a capillary bed in the lower hypothalamus, and the hypothalamic hormones that are destined to go to the anterior pituitary will be secreted into that capillary blood. The blood from these capillaries will drain into these hypothalamic hypophyseal portal veins. And portal veins, remember, are defined as veins between two capillary beds without returning back to the heart or the lungs, which is unusual. We've only seen two places that have them. The hypothalamic hypophyseal portal veins branch again into another series of capillaries within the anterior pituitary. These capillaries within the anterior pituitary are what will carry the hormones that are secreted by that gland, and they coalesce into veins that drain into the systemic venous blood. Those veins also collect capillary blood from the posterior pituitary gland. Now, why do we make such a special fuss about this whole unique connection? And it's because the utility of this unconventional vascular system is that the minute quantities, teeny tiny little quantities of hypothalamic hormones are carried in a highly concentrated form directly from the hypothalamus to their target cells in the anterior pituitary without being diluted at all in the systemic circulation. I just pretty much went through this whole hypothalamic portal vein connection and how they branch. So let's look at it on a picture. And here you see it. Once again, you have the hypophyseal artery that's ramifying in a capillary bed in the lower hypothalamus. And this will be where the hypothalamic hormones will jump in and go to the anterior pituitary. And blood from these capillaries are going to drain into this hypothalamic hypophyseal portal vein, okay, that you see right there in between, or the connection between those two capillary beds, where it's going from the red capillary bed we just discussed now to this blue capillary bed. So this blue capillary bed, depicted within the anterior pituitary, is what's carrying the hormones that were secreted by that gland, okay? And you see them coalescing, and then they form that anterior hypophyseal vein. And this is what drains into the systemic venous blood, okay? And that's collecting blood from both the anterior pituitary and also blood from the posterior pituitary goes into that hypophyseal vein, which will then lead into the systemic circulation. So if our pituitary gland is our king or queen of our endocrine system, the hypothalamus is the power behind the throne. The neurosecretory neurons of the hypothalamus secrete hormones that strictly control secretion of hormones from the anterior pituitary. The hypothalamic hormones are referred to as releasing hormones and inhibiting hormones, which reflects their influence on the anterior pituitary hormones. Hypothalamic releasing and inhibiting hormones are carried directly into the anterior pituitary gland via that hypothalamic hypophyseal portal vein we just discussed and saw a picture of. Specific hypothalamic hormones will bind to receptors on specific anterior pituitary cells and modulate the release of the hormone that they produce. And thus we refer to this as the. Now, it's also really important to remember that the hypothalamus, while very small, plays a crucial role in many important functions, including non-endocrine functions such as temperature regulation, regulation of the autonomic nervous system, and control of our appetite. We've already talked a little bit about the pulsatile secretion of hormones, such as with the LH surge. But it's a good thing to remind ourselves here that most hormones are released in a pulsatile secretion pattern. Pulsatile secretion is a biochemical phenomenon observed in a variety of cell types. Pulsatile activity is critical to the function of many hormones in order to maintain the delicate homeostatic balance necessary for essential life processes such as development and reproduction. Pulsatility is also thought to function to sensitize target tissues to the hormone of interest and upregulate receptors, which leads to improved responses. The hypothalamic pituitary adrenal axis, or HPA axis, is a complex set of direct influences and feedback interactions. The HPA axis is a major neuroendocrine system that controls reactions to stress and regulates many body processes, including digestion, mood and emotion. The hypothalamus is in charge of the stress response in our body, and we'll look at that when we get to our last module. Just know for now that the stress response is triggered and sends signals to both the pituitary gland and the adrenal medulla. And the basic output for the stress response is that our adrenal medulla will send out a release of glucocorticoids. And we previously examined how glucocorticoids also help us with halting the release of arachidonic acid from our cell membranes to make pro-inflammatory mediators. Now here is a very busy slide in your book, which I don't particularly find helpful, but you might. So you can just see that across the top you have all of the releasing and inhibiting hormones that the hypothalamus produces. And you see green lines for stimulating or releasing hormones and red lines for those that are inhibiting. And you can see whether they travel out through the hypophyseal portal system and make it to the anterior pituitary. Then you see the anterior pituitary hormones that are released, and these will be typically called stimulating hormones. Follicle stimulating, luteinizing hormone, growth hormone, TSH or thyroid stimulating hormone, prolactin, and ACTH. These then go out into the systemic circulation to the target organs, various organs like the thyroid gland or the adrenal cortex. And there we have some actively mediating hormones at the target organ, ultimate target organ level. Things like T3 and T4 that act on every cell in the body to stimulate metabolism. And here is another picture of the negative feedback control of the hypothalamus, pituitary, and thyroid axis that we went over previously. I went over it on the other slide just because I like the drawing better. I think like this is a beautiful fancy drawing, but I like the straight up and down for trying to learn the hormones and the negative feedback control. I think it's a little bit cleaner and easier to see, so that's why I put the other one up front. Okay, and this is just another slide to remind you of the pulsatile function of hormone release. And part of the pulsatile release is also related to our circadian rhythms. The levels of several hormones fluctuate according to the light and dark cycle and are also affected by sleep, feeding, and general behavior. The regulation and metabolism of several hormones are influenced by interactions between the effects of sleep and the intrinsic circadian system. These are growth hormone, melatonin, cortisol, leptin, and ghrelin. Then, not surprisingly, sleep disturbance, which negatively impacts hormonal rhythms and metabolism, is also associated with obesity, insulin insensitivity, diabetes, hormonal imbalance, and appetite dysregulation. Circadian disruption, typically induced by shift work, nighttime workers, may negatively impact the health of these workers due to things like impaired glucose and lipid homeostasis, reversed melatonin and cortisol rhythms, and loss of. We spent most of our time talking about the adenohypohysis. Let's talk a little bit about the neurohypophysis. The neurohypophysis is also known as the pars nervosa, or simply the posterior pituitary. So much easier to say, I keep tripping it up. Anatomists distinguish between three areas of this organ, starting with the closest to the hypothalamus, which is the median eminence. And then there is the infundibular stalk and the infundibular process, which forms the bulk of the posterior pituitary that we're always talking about. So let's look at a picture here. Okay, so we see the neurohypophysis as related to the hypothalamus and the important nuclei up in the hypothalamus, such as the supraoptic nucleus, which is a sort of an area of neurosecretory cells in the hypothalamus. And it's these cell bodies that will produce the peptide hormone vasopressin, which you also know as antidiuretic hormone. And next to that, you see the paraventricular nucleus in the hypothalamus, which is a group of neurons. And these PVN neurons project directly to the posterior pituitary, where they will release oxytocin into the general circulation. Sorry, I'm coughing here. My neighbor is out mowing grass, and I'm allergic to the grass pollens. I don't have COVID, I hope. Okay, so the hormones ADH and oxytocin, which are produced in the hypothalamus, will then travel down the axons to the neurohypophysis, which will serve as a storage and release site for the hormones. So vasopressin, or antidiuretic hormone, or arginine vasopressin, known by many names, you know it very well. It helps our body to conserve water. It increases the amount of solute-free water that's reabsorbed back into circulation from the filtrate in the kidney tubules of the nephrons, and it also constricts the arterioles, which will increase peripheral vascular resistance and raise the arterial blood pressure. The other hormone is oxytocin. And oxytocin has at least two primary actions. It causes milk ejection from the lactating breast, which is regulated by a positive feedback mechanism. This is the milk letdown reflex, okay? And try to keep oxytocin and prolactin straight. Prolactin stimulates the breast development and the formation of the milk, but oxytocin and stimulation of the nipple actually causes the milk to be let down or ejected. Oxytocin also stimulates the contraction of the uterine muscles that occurs during and after childbirth, which is regulated also by a positive feedback mechanism. It's also said to help with bonding with the baby as well. So oxytocin is important for cervical dilation before birth, and it causes contractions during the second and third stages of labor. And when labor is not progressing, we'll give people IV Pitocin. Pitocin is the synthetic form of oxytocin. So oxytocin is controlled by a positive feedback mechanism where the release of the hormone causes an action that stimulates more of its own release. And so when contraction of the uterus starts, oxytocin gets released, which stimulates more contractions and more oxytocin to be released. And you can see why then we also will help labor by giving Pitocin. the synthetic form of oxytocin, to assist with those contractions when labor is not progressing. The pineal gland is so named because it is a pine nut shaped structure that's located on the dorsal aspect of the brain's diencephalon. The pineal gland is also known as the epiphysis, and it synthesizes and secretes melatonin, a structurally simple hormone that communicates information about the environmental lighting to various parts of the body. Ultimately, melatonin has the ability to entrain biological rhythms and has important effects on reproductive function of many animals. The light transducing ability of the pineal gland has led some to call the pineal gland the third eye. Histologically, the pineal gland is composed of pinealocytes and glial cells. Light exposure on the retina is relayed to the suprachiasmatic nucleus of the hypothalamus in an area of the brain that's well known to coordinate biological clock signals. Fibers from the hypothalamus then descend to the spinal cord and ultimately project to the superior cervical ganglia, from which postganglionic neurons will ascend back to the pineal gland. In this way, the pineal gland is similar to the adrenal medulla in that it senses and transduces signals from the sympathetic nerve system into a hormonal signal. The precursor to the hormone melatonin is the neurotransmitter serotonin, another neuroendocrine connection there. The neurotransmitter that itself, serotonin, is derived also from the amino acid tryptophan. Within the pineal gland, serotonin is acetylated and then later methylated to yield melatonin. The synthesis and secretion of melatonin is dramatically affected by light exposure to the eyes. The fundamental pattern observed is that serum concentrations of melatonin are low during the daylight hours and increases to a peak during dark or nighttime hours. And as a note, I once heard an expert who was lecturing on the use of melatonin. And those of you who have sleep problems, most people go to GNC and grab a bottle of melatonin and they take it wrong. And just like with NyQuil and aspirin and ibuprofen, how we always tend to think that more is better, more is really not better with melatonin, and taking too much melatonin might actually backfire on you. So be very careful in doing over-the-counter dosing of melatonin and stick to the recommended dosage. Okay, now let's just look at some learning objectives for the second half of this lecture. We're going to look at structures, locations, functions, and hormones of the thyroid and parathyroid glands, although we have looked at the thyroid quite a bit already. We're going to discuss the structure, location, and functions of the adrenal glands. God, I can't talk today. And we're going to compare and contrast the functions of the hormones that are produced by the cells of the adrenal cortex and those secreted by the adrenal medulla. Then we are going to describe the cells found in the pancreatic islets and their hormonal secretions. We're going to identify those hormones' functions, and then we'll briefly look at some of the functions of the ovaries and testes, but most of that will be left to our next section on reproduction. And similarly with the placenta, we're not going to look at that too much because that should go over with the reproductive aspects. And we've pretty much already talked about the thymus, the GI tract, and all that, but we're just going to lightly touch on how these relate to, we're functioning as endocrine organs themselves. Okay, and then we'll examine a couple of endocrine disorders. I already said we'll touch on gigantism and acromegaly. And of course, we have to talk about diabetes, right? Okay, so let's start with the thyroid gland. So if our pituitary is our master or king and queen gland, and the hypothalamus is the power behind the throne, our thyroid gland is the James Brown of the endocrine system. It is the hardest working gland in the endocrine business. Not a cell in your body goes untouched by the effects of the thyroid gland. It's made up of two large lateral lobes and is connected by a narrow isthmus, which you'll see on the next page or next slide. And it has a worm-like projection of thyroid tissue that's called the pyramidal lobe that extends upward from the isthmus. In an adult, it weighs about 30 grams, and we talk about it as the butterfly in the neck. It's the butterfly-shaped organ. So let's look at that thyroid gland. Okay, so you can see that little worm-like projection upward, or the pyramidal lobe. You can see the lobulated thyroid tissue running between the carotids and the jugulars, and it kind of overlies the trachea, right? You can see those C rings of the trachea below it. And we often ask people to swallow when we're checking their thyroid so that we can kind of feel the movement of the gland. Is it mobile? Is it, large? Or is it, if it's too large, having a goiter? The thyroid gland is unusual in that the hormones are stored in cavities that are surrounded by secretory cells, which make up a follicle. And you see the follicles right here. The left-hand side is a drawing, and the right-hand side is a preserved specimen. To secrete the hormone, the thyroid hormone is reabsorbed from the cavity and then released into the surrounding interstitial spaces. The stored hormone is bound to a glycoprotein, and this stored hormone is called colloid, which is that big globular pink staining that you see in the center of the follicle. This gland secretes iodine-containing hormones that are called triiodothyronine, or T3, and thyroxine, T4, of which T3 is the more active form, but you have it in much smaller amounts. T4 is really a prohormone that will change into T3. The thyroid hormones will regulate the basal metabolic rate, and it is regulated by the pituitary hormone, TSH, thyroid stimulating hormone. The thyroid gland also secretes calcitonin, which regulates blood calcium levels. Secretion of calcitonin causes blood. Thyroid hormones are derivatives of the amino acid tyrosine, covalently bound to iodine. The two principal thyroid hormones are thyroxine, or T4, tetraiodothyronine, or triiodothyronine, T3. Most of the thyroid hormone secreted from the thyroid gland is T4, but T3 is considerably more active. And the bulk of the T3 is derived by deiodination of T4 in peripheral tissues, especially in the liver and kidney. Deiodination of T4 also will yield a reverse T3, which is a molecule that we don't know has any metabolic activity. The thyroid hormones are poorly soluble in water, and therefore more than 99% of the T3 and T4 that are circulating in blood are bound to carrier proteins under the same principles that we've learned previously with cholesterol, right? The principal carrier of thyroid hormones is called thyroxine binding globulin, TBG, okay? And these are glycoproteins that are synthesized in the liver. Two other important carriers are transthyretin and albumin. Carrier proteins will allow a maintenance of a stable pool of thyroid hormones from which the active free hormones will be released for uptake at the target cells. Calcitonin is a thyroid hormone that's known to participate in calcium and phosphorus metabolism. The major source of calcitonin is from the parafollicular, or C cells, in the thyroid gland. And don't start to think that parafollicular means it's from the parathyroid, okay? Parafollicular C cells are in the thyroid gland. Calcitonin has the ability to decrease blood calcium levels. One way it does this is by suppressing resorption of bone by inhibiting the activity of osteoclasts, the cell that is known for digesting the bone matrix and releasing calcium and phosphorus into the blood. As well, it acts at the kidney. Calcium and phosphorus are prevented from being lost in the urine by reabsorption in the kidney tubules. Calcitonin inhibits tubular reabsorption of these two ions, leading to increased rates of their loss in the urine. The most prominent factor controlling calcitonin secretion is the extracellular concentration of ionized calcium. elevated blood calcium levels will strongly stimulate calcitonin secretion, and secretion is suppressed when calcium concentration is low. Also controlling calcium concentrations is parathyroid hormone. Parathyroid hormone is the most important endocrine regulator of calcium and phosphorus concentration in extracellular fluid. Parathyroid hormone is secreted from cells of the parathyroid glands, and it finds its major target in cells in the bone and kidney. And you'll see where it gets its name when we look at our next slide, how these parathyroid glands are like little islands or glands that are embedded in the posterior surface of the thyroid's lateral lobes. And you can imagine that when you have to have thyroid surgery to remove parts of the thyroid gland, say if you had thyroid cancer, you're probably going to take a lot of that parathyroid tissue with you. But what we do in surgical suites is we try to have someone over there also cutting out the parathyroid glands, and we reimplant them to help maintain calcium homeostasis after a big thyroid surgery. We implant them in people's left arm usually, so we know to look for the scar over there. Parathyroid hormone is secreted primarily by the chief cells of the parathyroid glands in response to low extracellular concentrations of free calcium. And it attempts to bring back the calcium concentration to the normal range by three processes. First, it mobilizes calcium from bone. The mechanisms are kind of obscure, but one of the effects of parathyroid hormone is to stimulate the osteoclasts to reabsorb bone mineral by liberating calcium into the blood. It also will indirectly stimulate the production of the active form of vitamin D in the kidney, and vitamin D will induce synthesis of a calcium binding protein in the intestinal epithelial cells that will then facilitate efficient absorption of calcium into the body. It does this by stimulating tubular reabsorption of calcium. Another effect of parathyroid hormone on the kidney is also to stimulate the loss of phosphate ions in the urine. Okay, so let's look at this important parathyroid gland. And you see there are about 4 to 5 parathyroid glands that are located on the back of the thyroid gland in variable locations. Again, using food, they're about the size of a lentil seed. And they aren't visible or felt when you examine the thyroid gland by doing a neck exam. And here's a histological section of parathyroid tissue. And half of this slide is thyroid tissue and thyroid follicles. And typically you'll see the parathyroid gland contains a large amount of adipose tissue, which expands in volume beginning at puberty. The PTH producing chief cells are numerous, with central nuclei and pale cytoplasm. The more active the cells are in producing and secreting PTH, the darker their cytoplasm will appear. There's also usually oxyphil cells who have an unknown function that you can see, but this is pretty low magnification, so you'd really have to look at this at a much higher magnification up close to determine and see all that. And we've already gone through how parathyroid mobilizes calcium from the bone, and it does so by indirectly stimulating the osteoclasts. The osteoclasts themselves don't have a receptor for parathyroid hormone, but parathyroid hormone will bind to osteoblasts, the cells that are responsible for creating bone, and then these will stimulate the osteoclast precursors to fuse and form new osteoclasts that will then start destroying the bone and releasing the calcium. As well, we talked about how parathyroid hormone will cause the kidney to increase absorption of calcium at the distal convoluted tubule and collecting ducts, which you should know well from just having done the kidney. As well, it will also inhibit phosphate reabsorption that will result in a decrease in plasma phosphate concentration. And normally, phosphate will complex with calcium, and therefore when you decrease the phosphate concentration, it will cause an increase in the ionized calcium in the blood. And finally, we talked about how parathyroid hormone will stimulate the conversion of inactive 25-hydroxyvitamin D into its active form, the 1,25-dihydroxyvitamin D form, which is called calcitriol. And this is then released into circulation. And that active form of vitamin D then induces a synthesis of a calcium-binding protein in the intestinal epithelial cells that will facilitate the absorption of calcium. All right, and here is a nice slide that overall gives us a view of the regulation of calcium levels in the blood. And plenty of med students get obsessed about having to learn how to calculate their ionized calcium and the levels of free calcium, and it's really important because low levels of free calcium can cause your heart rate to slow down or speed up. It can cause you to have muscle spasms and even go into a coma. So calcium homeostasis is really important. The adrenal glands, technically called the suprarenal glands, are endocrine glands that produce a variety of hormones, including adrenaline and the steroids aldosterone and cortisol. They're found at the top of each kidney. They're like a cap on each kidney. And each gland has an outer cortex which produces steroid hormones and an inner medulla. And that's composed of neurosecretory tissue. And we'll look at that. Let's here look at the location of the adrenal gland, and you see it sitting up there at the top of the kidney on the right side of the body. When you think about the adrenal glands, think about stress. Stress takes many forms, such as taking the exams in this class, especially under the conditions of COVID-19. Or speaking of COVID-19, recovering from a major illness is a stress on the body. Having to maintain your levels of energy in the face of starvation is a stress on the body. So there's all different kinds of forms that stress can take, and we'll talk about that a little bit more at the end of the semester. So your adrenal glands will produce 3 major classes of hormones, each of which will aid in dealing with the multitude of small and large stresses that you face daily. There's no doubt that at least 2 of these groups, the glucocorticoids and the mineralocorticoids. An adrenal gland that is sectioned has 2 distinct regions: the inner medulla, which is the source of catecholamines such as epinephrine and norepinephrine, and the outer cortex that secretes several classes of steroid hormones, the glucocorticoids and the mineralocorticoids. And histologic examination of the cortex will reveal 3 concentric zones of cells that differ in the major steroid hormones that they secrete. The adrenal gland is really 2 glands in one. Sectioning through the adrenal gland, you'll see a pale medulla that's surrounded by a darker cortex, and these 2 regions will produce distinctly different groups of hormones. And then you see the adrenal itself is covered in a capsule. So now we'll look up close at the cortex, which has these 3 different zones. The outer zone, the zona glomerulosa, the middle zone, the zona fasciculata, and then the inner zone, the zona reticularis, which abuts the medulla. And here we have an up-close histological section of those 3 different layers. Here you see the outermost zone is the zona glomerulosa, and cells within this zone are more columnar in shape and are arranged in irregular cords. Sometimes they're described as being in arcades. The zona fasciculata is the middle and largest of the 3 zones in the cortex. Cells in the fasciculata are polyhedral and usually have a foamy appearance due to abundant lipid droplets. And the innermost zone is the zona reticularis, and cells in this zone are arranged in cords that project in many different directions and anastomose with one another. And the medulla is populated with large columnar cells called chromaffin cells, which synthesize and secrete catecholamines. Ganglion cells are also observed, but infrequently. Blood from throughout the adrenal gland collects into large medullary veins that exit the gland. Here, we are reminded that the adrenal cortex is partitioned into 3 concentric zones of steroid-synthesizing cells: the glomerulosa, which produces the mineralocorticoids, represented best by aldosterone; the fasciculata, which produces the glucocorticoids, best represented by cortisol; and the zona reticularis, which produces the gonadocorticoids. The mineralocorticoids are secreted by the outermost region of the aptly named mineralocorticoids play a critical role in regulating the concentration of minerals, particularly sodium and potassium, in extracellular fluids. Loss of these hormones leads to a rapidly life-threatening abnormality in electrolyte and fluid balance. The major target of aldosterone is the distal tubule of the kidney, where it stimulates exchange of sodium and potassium. 3 primary physiological effects of aldosterone will result, which is the increased reabsorption of sodium. Sodium lost in the urine is decreased under stimulation from aldosterone. It also increases the resorption of water, with consequent expansion of the extracellular fluid volume. This is an osmotic effect that's directly related to the increased resorption of sodium, and increased renal excretion of potassium. And you know these already because we just went over the kidney. The glucocorticoids are secreted by the middle region of the adrenal cortex, the fasciculata. Cortisol is the principal glucocorticoid, and it will increase blood glucose levels and reduce inflammation. Loss of glucocorticoid secretion will lead to a state of deranged metabolism and an inability to deal with stressors, which, if left untreated, will be fatal. Glucocorticoids are among our most frequently used drugs, and they are not benign as many people start to think they are. They're often prescribed for their anti-inflammatory and immunosuppressive properties, and we're prescribing more and more of them because of the opiate crisis, but I presume one day we'll also see the sad effects of prescribing too many glucocorticoids in response to this. The vast majority of glucocorticoid activity is from cortisol, also known as hydrocortisone, and will be prescribed to you as prednisone. Corticosterone is the other major glucocorticoid that you'll hear about. The name glucocorticoid derives from early observations that these hormones were involved in glucose metabolism. During the fasting state, cortisol stimulates several processes that collectively serve to increase and maintain the normal concentration of glucose in the blood. These effects will include stimulation of gluconeogenesis, particularly in the liver. It will also mobilize amino acids from extrahepatic tissues that will serve as substrates for gluconeogenesis. It will inhibit the uptake of glucose in the muscle and adipose tissue in order to conserve glucose. And cortisol will also stimulate fat breakdown in adipose tissues. The fatty acids that will be released by lipolysis are then used for the production of energy in tissues like muscle. And the released glycerol will provide another substrate for gluconeogenesis. And I should also address the potent anti-inflammatory and immunosuppressive properties of the glucocorticoids. This is particularly evident when we administer them in pharmacologic doses. But it's also important for our normal immune responses. As a consequence, glucocorticoids such as prednisone are widely used as drugs to treat inflammatory conditions such as arthritis, dermatitis. We also use them as adjunct therapy for autoimmune diseases. And I think I'm going to stop here because we're now over 1 hour, and we'll pick up with the secretion of cortisol slide, and I'll do that at another time. Okay? Hope you're all doing well and staying safe, secure, and happy.